So, what we're doing, weapons? And my son was also here at home, and he asked me, did you get homework? I said, yeah, they're going to give exam. That's someone. Okay. So I know this thing happens. Don't worry about it. Yeah, okay.
So we were talking about the Maya tone and specifically what goes into it and how PAX1 activates SOX 9, which activates SOX 5 and 6 to lead to the formation of cartilage. And then we talked about how the Senditone has a slight amount of sonic hedgehog due to its placement. So the PAX1 is within the sclerotome, and that's how it allows it to get cartilage, and then the Senditone has a slight amount of sonic hedgehog. And to give or to help the tendons, we'll get to that. And then the myotome starts secreting the FGF when it starts moving down. So there's a high concentration at the top, and it's lower as it moves down. And then the FGF, which are the cells beneath the myotome, will express P3 and ERM1 to induce expression of the
P1.
P1.
Yeah, it was P1.
Oh. P1 and ERM1 to induce expression of the scleral axis to induce tendon formation. And then because
P1, I don't P3 there, it should be P1.
And then because sonic hedgehog is excessively present in the sclerotome, it can block FGF signaling to not become the tendon. And then sonic hedgehog and SOX 5 and 6 will block FGF as well to stay cartilage in the sclerotome. And then we talked about the myoblast to muscle fiber.
Okay.
So instead of the myosome cells produce margin everywhere, there are more cells called myoblasts, which muscle cells, and they don't work as individual cells. The myoblasts piece together to form nucleated muscle fiber. And then in mice, the total number of muscle fibers in the first week, they grow by the addition of myoblasts after several weeks, and muscle fiber increases in size one.
Yeah.
And then increasing the size of growth which happens in cells. And we talked about myoblasts and muscle fiber. Myoblasts should come out of the cell cycle precursor cells. So sonic hedgehog will be expressed in NYOD or NYFS. And the fourth process is called myotome cells, and not divide and then multiply to form myoblasts, and then effects of the stats and and then the myoblasts secrete photometric unnecessary matrix and amplify beta
Okay, then what happens?
Then the alignment of the myoblasts in chain. So the myoblasts start expressing muscle-specific cadherin or M-cadherin and other M-cams, which are muscle cell adhesion molecules. And then membrane fusion. Calcium is important for fusion, and the calcium enters into the myoblasts and cause membrane fusion. There's also meltrin, which aids in membrane fusion, and then the M-cadherin.
So American is like isomer, right? We have isomer here on the sperms. Here it's American. Okay, then what happens?
Then lineage specification occurs. PAX3 activates MyoD or MyoF5.
Wait, is that supposed to be PAX 1 too? Or the
PAX3.
It's PAX 3 now?
It was
Yeah, PAX1 was in the cartilage, right?
Yeah.
In the muscle, it's PAX3.
and then to the myoblasts. They then start myogen expression and activation of muscle-specific genes. And then stabilization of the membrane is sealing everything. Myofurlin and dysferlin. Dysferlin are sealing proteins, and they stabilize the membrane phospholipids. And then growth of the myofiber. They secrete interleukin 4, IL-4, which attracts other myoblasts to come and fuse. And then regulation of the myofiber. myostain secretes by muscle fiber, is a negative regulator, and mutation in myostatin can cause herculine, which is having a lot of muscle mass, and they fuse within. Oh, sorry.
Okay.
Then we talked about satellite cells. So unfused muscle progenitors in the dermamyotome, they express PAX3 and PAX7, which together inhibit MyoD. And then also microRNA 489, which is a post-transcription regulator, which binds to the mRNA of MYT5 to inhibit translation. And then these can have symmetric division with both expressing PAX3, PAX7, and microRNA 489, or asymmetric with one expressing all three and the other only expressing PAX3 and PAX7, which would then allow the expression of MYT5 and form myoblasts.
Okay. So we have talked about formation of muscle. We have talked about formation of cartilage. We have talked about formation of tendon, right? So now bone. So there are three. So we are talking about bone formation. And bone formation is called osteogenesis and also called ossification. Also called ossification. Okay. So the bone can come from three distinct lineages. So there can be three distinct lineages from which bone can come. Three different places the bone can come. Number one is paraxial mesoderm. So paraxial mesoderm can give us the bone, and it can give you vertebral bones. So all the vertebrae that is coming is from the paraxial mesoderm. And it can also give you the craniofacial bones. Craniofacial means our cranium head and the face bone. Okay? That is coming from the paraxial. Now we are saying vertebral bone, that means the ribs are also coming from there, because ribs are attached to the vertebral arteries, right? So ribs are also coming from there. And then we have that other mesoderm, what was lateral plate mesoderm. Lateral plate mesoderm was giving us the limb bones. So they are coming from here. Lateral plate mesoderm is going to give you limb bone. Now, there's one more thing called neural crest cell. Remember, neural crest was there. We said neural crest gives so many things. So one of the things that neural crest provide is some of the facial bones are made from neural crest. And also some cartilage also from there, but we're talking about the bones. So these are the three places from where different types of bones are formed. Okay? Now, how this bone is formed, we're going to talk about here. So all this is mesoderm, except neural crest, right? Neural crest is not mesoderm, but we're talking about lateral plate mesoderm, we're talking about paraxial mesoderm. Now, so if this is mesoderm, right, there are these cells there. So these cells which are making mesoderm, we call them mesenchyme cells. Okay? So the mesenchyme cells, so all the bone is coming from mesenchyme cells. Those are making mesoderm. So what can happen? The mesenchyme mesenchyme cell can directly transform into bone. They can directly transform into bone, directly change into bone. And this is called, this type of bone formation is called intramembranous ossification. So, mesenchyme cells are there. These mesenchyme cells directly change into a bone. Okay, so this is called intramembranous ossification. Or the other thing what can happen is, the second thing, that mesenchyme cells, they change to cartilage. They change to cartilage. Okay. And then what happened? The cartilage is replaced by bone. So first cartilage is formed, and then cartilage is replaced by bone. And this process is called endochordal ossification. So what is happening? So we are saying bone formation can occur in 2 ways. We said there can be 3 lineages, right? You can have retrograde mesoderm giving you limb vein. You can have praxillary mesoderm giving you vertebrae and the ribs and craniofacial bone. Then you can have neural crests that give you the facial bone. And then how the bone is formed. We can say that mesenchyme cell, they can directly transform into bone, and that is called intramembranous ossification. In the other process, what can happen, that mesenchyme cell, they change to cartilage, and that cartilage is replaced by bone, and this process is called endochordal ossification. Okay? So we are going to talk in detail about endochordal ossification because most of the bones are formed by this process. Okay? So you can talk to each other and see if there is any doubt about this process. Any doubt?
No.
Okay, now. So, endochordal ossification. Endochordal ossification. So, in endochordal ossification, all the ribs, vertebrae, and limbs, they are all coming through this process of endochordal ossification. Now these vertebrae and ribs, they are coming from somites. Remember, we did talk about that somite. We said Hox gene play a role there, and we said each Hox gene pair can tell the types of vertebrae there. And we said mesenchyme is split into the somite, again the Hox. So that's why these vertebrae are from the somites. And then limbs, these are coming from lateral plate mesoderm. So how this process occurs? This is a complex process, and the endochondral ossification, it can be divided into 5 steps. And we'll talk about these steps. It can be divided into 5 steps. Now, we have the step 1 and 2. That's what we're going to talk about. Step 1 and 2 is a process what is called commitment, and compaction. So that's what happened in step 1 and 2, commitment and compaction. So what happened that in the, for example, this is the mesoderm, okay? In this mesoderm, say, okay, this is a somite, and these cells, they say, okay, we want to be bone, right? So what these cells do, these cells which are going to become the bone, we call them the mesenchyme cells commit to become bone. That's what their first thing is. They commit. Now, when they say they commit to form bone, they are not going to form the bone itself directly, right? We said they need to form cartilage. So what happened? These cells are kind of separated like this. So this is each of these cells is there. And when you're saying they commit to become bone, they commit to become bone in a way that the cell, the inner cells, they commit to become cartilage. Inner cells commit to become cartilage. And the outer cells, the cells which are on the outside, the outer cells, they commit to become bone. Outer cells commit to become bone. And what happened, that this is the commitment. And then what happened, these cells which are separated so far away, they actually compact. The compaction takes place, and these cells, they all compact in this structure like this. They form this structure, this is called compact nodule. So mesenchyme cell, they first commit, they said, okay, the inner cells are going to become cartilage, outer cells are going to become the bone, and then they should do this process compaction, and they essentially form a compact nodule. Now, this process, in this process of bone formation, here, the bone morphogenic factor, BMP, is critical. Now this is interesting. Before this, when the muscle was forming, when the syndrome was forming, when the cartilage was forming, we were inhibiting the BMP. Remember? We said no organism, but now we don't want to inhibit. So this means that bone formation takes place after the muscle and the cartilage are being demolished. So now what is happening? Now the BMP is critical. What BMP does? BMP causes two things on these cells. Number one, cause expression of N-cadherin. On the cells. Now, N-cadherin is used with neural cadherin, right? But these cells express the same cadherin as in the neural cell. Okay? So these cells right here, so BMP coming there, so say this one like this, commitment took place, BMP come there, BMP says, okay, let's these cells start expressing N-cadherin. So this is all N-cadherin. So N-cadherin is going to make those cells stick together, right? So these cells are kind of like sticking together with N-cadherin. So N-cadherin is there, N-cadherin is causing them to stick together. Now, second what it does is, this is the first thing. Second it does is cause expression of NCAM. NCAM. Neural cell adhesion molecule. Okay, NCAM. Now, these cells also start expressing NCAM. So you have now the NCAM also. So this NCAM is there. What NCAM does, NCAM retain these cells in the compact nodule. So NCAM stabilize the compact nodule structure. Making sense? So N-cadherin cause those to compact and NCAM maintain that compact structure. So two things are happening. Now the third thing it does is, this is all coming from the BMP. BMP is done N-cadherin, BMP is doing NCAM. The third thing BMP does is it induces expression of SOX9. So SOX9 is a transcription factor, and what SOX9 does? SOX9, it activates transcription of collagen of collagen 2. There are many types of collagen there, collagen 2. And aggrecan. Collagen 2 and aggrecan. Now, these two are critical for cartilage. Remember, we said this is endochordal ossification. So you have to become cartilage first, and then cartilage should be replaced by the bone, right? And this SOX9 is activating transcription of collagen and aggrecan, which is responsible for cartilage function. Now, this is actually critical. So mutations in SOX9 in human, they cause a disease, what we call camptomeric dysplasia. Camptomeric dysplasia. Now, this is a rare disorder. This is a rare disorder, but this rare disorder is observed in the skeletal development. So the babies, most of babies with this syndrome, they actually die by respiratory failure. They die by respiratory failure. Why do you think that should happen? Say it again?
Does it have to do something with the ribs?
Yes, they have to do with the ribs. The rib cartilage will not function, right? What is the other thing? That's a very good answer. So rib cartilage is not good. So ribs need to expand, right? That's the first thing. What is the other reason? So this is your windpipe right here. It come here and you have your lungs here. Windpipe is called trachea. These are lungs. Trachea changed to bronchus and bronchioles and all those things, right? The trachea structure is maintained, and again here the bronchiole, these are bronchiole. These are the bronchiole. Trachea, bronchiole, right? So each of this is actually this structure. This is kind of a polythene, right? Very filmy structure. But these need to be maintained, and that tube should be maintained, and right here is the cartilage. So that cartilage is maintaining it. So then if that is not there, you have trachea, bronchiole, bronchiole, bronchioles, everything will just collapse. No air will go in. So these kids usually die. So what we are saying? We are saying that endocardial ossification is a multi-step process, 5-step process, and we started talking about step 1 and step 2. We said step 1 and step 2 are commitment and compaction. These cells become committed to become the inner cell committed to become the cartilage, and outer cell committed to become the bone. And when that happens after commitment, the BMP play a role. BMP is secreted. What BMP does, BMP start expressing and adhering on these cells. That cause compaction of these cells. And then BMP also start expressing and CAM on these cells so that this structure is maintained. And this process is called compaction, and the structure which is formed is called compact nodule. Okay. Now, then what happened that BMP also start expressing, tell these cells to start expressing SOX9. SOX9 is a transcription factor. That SOX9 causes transcription of collagen 2 and aggrecan, and these are responsible for the function of the cartilage. And mutations, rare mutation in SOX9 can cause camptometric dysplasia, and these kids usually die by a respiratory failure. Of course, they will have other problems as well for the bone development, but when they die, then what can you say, right? So far it is okay? So talk about these 2 steps, then we'll move on.
So whenever the cells become the bone cells, whatever, do they start producing again?
No, BMP is coming from the sound. Another vector will also submit for the right? So that means by the time the other factors like protein production is going down, right? And we said like there are some epiglottis cell attached there could be problems. So that means that is going down. So it's all time dependent. Any question here? Okay, now we are at step number 3. 3 and 4. Okay. And that is proliferation and growth. So what happened that now you have these cells. This was your compact nodule. This cell now, as we say, proliferation. It means they start dividing very fast. Okay? So these compact nodule cells, compact nodule cells, they differentiate to chondrocytes. So they differentiate, it means they are no more compact nodule cells, they are no more mesenchyme cells. What they become, they are still there, now they become chondrocytes. Right? So first they differentiate into chondrocytes. And what they happen, these chondrocytes, they divide rapidly. So they start dividing like crazy. And when they are dividing, while dividing, they secrete cartilage specific extracellular matrix. They secrete cartilage specific extracellular matrix. Now, and these cells form a cartilage model of the bone. They form a cartilage model of the bone. So what happened, that these cells are dividing, they divide a lot, and ultimately they form a structure like this, in which you have all these chondrocytes are divided. Okay, so they have divided, and what they have formed a, so these are proliferating chondrocytes, and what they have done, they have made a cartilage model of bone. So they make a cartilage model of bone. Now what they do? So outermost cell, these outermost cells, which are in this bone model, okay, they secrete a matrix around this. So this is the matrix. This is kind of matrix. So outermost cell of this model, of this bone model, okay, secrete a matrix called perichondrium. So now this matrix is secreted, this is perichondrium. Okay? Now what happened? And then you have some cells which are still outside of this. the cells here are there. So the cells which are there outside, and these cells are also surrounding somewhere inside also like this. Throughout. Throughout there will be. These cells, the blue cells, these will be called perichondriocytes. So what is happening? We are saying that these compact nodule cells, they differentiate into chondriocytes. These chondriocytes divide rapidly. While they are dividing, they are secreting the cartilage-specific extracellular matrix around them. And then these cells proliferate more and form a cartilage model of the bone. The outer cell of these, they actually start secreting an extracellular matrix that is called perichondrium. And then these cells, the innermost cell, the inner cell, they become the chondriocytes, and the surrounding cell here, these cells become perichondriocyte. Making sense? So the cells which are outer cell, they become perichondriocyte, and the inner cell, they become chondriocyte. Remember, we said right in the beginning, we said when they commit, we said the inner cell commit to become the cartilage, the outer cell commit to become the bone. And here we are saying the inner cells are called chondriocyte, the outer cells are called perichondriocyte. Making sense? Any doubt here so far? Okay, then what happened? So this is the proliferation, right? Then we are talking about the growth. Then what is happening? So all of a sudden what happens, the cells which are here in the center, so far every cell was divided, right? And then what happened, so this was the phase 3, the proliferation, now we are at the phase 4 and the growth. So fourth phase what happened, that the chondriocytes, they stop dividing. The chondriocytes stop dividing. And what happened when they stop dividing, they increase their volume. So these cells which were there, they now stop dividing and they increase the volume. So think about this. So the central cell, they were like that, and now they have become like this big. So they have increased their volume. They have become like that big. Okay? And again, this is the bone model. This is the bone model there. Say this is the bone model. And surrounding cells are still the small. There's some chondriocytes here. And then you have these cells. The perichondriocytes are there. So these cells have stopped dividing chondriocytes. And what they have done, they increase their volume. And when they increase their volume, we call them, they become hypertrophic. They become hypertrophic chondriocytes. Now, these hypertrophic hypertrophic chondrocytes are exceptionally important. Hypertrophic chondrocytes are exceptionally important for the rate at which a bone is going to grow. So think about that you have limb bone, right? You have this forelimb bone and hind limb bone. Hind limb bones are longer than the forelimb bone. So it means that forelimb bone will have the smaller hypertrophic chondrocytes versus hind limb bone, they will have bigger hypertrophic chondrocytes. So how big the size of this hypertrophic chondrocyte, that is going to determine how long a bone will be. Now, in the same organism, it can be like that. But think about in the mouse versus human. Mouse is very small. So in mouse, the hypertrophic chondrocyte size is very smaller than in the human versus giraffe. So same bone become very long versus very small. That is dependent on the hypertrophic chondrocytes. That's the critical thing. Now what happens? These hypertrophic chondrocytes are formed. So that is the growth phase is happening. Then what is happening? That these hypertrophic chondrocytes, they start secreting, they secrete two things, two major things here. Number one is collagen, collagen 10. They start secreting collagen 10. Okay? And then they also secrete, second thing is fibronectin. Now remember we said that calcium, you know that calcium is very important for bone formation. They said that the pregnant bone should take a lot of calcium, blah, blah, blah things. What is the reason? Because these bones need to be calcified. And that calcification occurred from calcium phosphate. And when you have collagen 10 and fibronectin secreted, that lead to calcification of the bone. Okay? So then, therefore, this lead to calcification of bone from calcium phosphate. So at that time, the calcium start depositing inside the bone. So far making sense? So we are saying that these chondrocytes, they stop dividing. That's the step number 4, growth. Chondrocytes stop dividing, and what they do, they increase their size. They become hypertrophic. And that hypertrophic determines how quickly, how longer a bone will be, or how short a bone will be. Then we are saying these hypertrophic chondrocytes, they start secreting collagen 10 and fibronectin. That causes the calcification of the bone via calcium phosphate, and here the calcium starts depositing in the bone. That was our step number 4, phase number 4. Now what happened? Now we are going at the phase 5, or step 5. Now, in the step 5, what we need to do is, we need to, this is cartilage, right? So we need to form the bone. And how the bone is going to form? So cartilage is formed, and also there is no blood yet. Remember that in the bone, what you have? In the bone, in the center you have the bone marrow. Bone marrow have all the hematopoietic stem cells which are going to give you all the blood, right? So you need to get those things in, and you need to form the bone as well. You're not cartilage, you're bone. So what happened here? Two things happened. So these hypertrophic cells, so calcium getting in, right? Now step 5. Hypertrophic chondrocytes start secreting VEGF. So now they are secreting VEGF. And what VEGF is going to do, VEGF is going to, what is the function of VEGF? We did talk when we were talking about trophic factor. When we're talking formation of trophic. Chorion, how the chorion is formed. We said that since cytotrophoblasts secrete VEGF, and what VEGF does, VEGF turns mesenchyme cells into the blood vessels. So the same thing happened there. Start secreting VEGF, that cause mesenchyme cells to make blood vessels. So, VEGF is secreted and attract the blood vessel, make blood vessel and attract blood vessels. So, here so they start secreting here the VEGF throughout. So this is VEGF. And that causes the blood vessels to move in. So from here, blood vessels start moving. That's the first thing it does. Okay. Now, second thing what it does. So, see, a lot of things these hypertrophic cells are doing. So these hypertrophic cells hypertrophic chondrocytes, they secrete Indian hedgehog. They secrete Indian hedgehog. And so this Indian hedgehog is secreted. So Indian hedgehog is secreted like this. Indian hedgehog is secreted like this. Indian hedgehog is secreted like this. So these blue are the perichondriocyte, right? So Indian hedgehog on perichondriocytes. So Indian hedgehog is secreted on the perichondriocyte, and then what it does, that cause expression of. So ultimately what we want bone to be formed, right? That cause expression of RUNX2 in perichondriocytes. And when that happens, these perichondriocytes change to osteoblast. So perichondriocytes change to osteoblast. Osteoblasts are the precursor of the osteocytes. Osteocytes are going to secrete the bone. Okay. So what is going to happen? Now these cells were perichondriocytes. Now what happened? These cells have changed into osteoblasts. So here, let me change their color. So these cells have now become the osteoblasts. So what happened? Perichondriocyte change to osteoblast. Now, what happened? These osteoblasts are there. These osteoblasts become responsive to wind. They become responsive to wind signaling. And when they become responsive to wind signaling, what they happen, they start become wind and make ostrix. And become osteocyte and start secreting bone matrix. So let me go back. So what is happening? What is happening? We are saying these hypertrophic chondrocytes, they make collagen, fibronectin, cause calcium phosphate to change into calcium, a lot of calcium there. Right? So it's all cartilage and the calcification calcium start coming in the body. Then we are saying these hypertrophic cells, they start making Indian hedgehog, secreting out Indian hedgehog. Indian hedgehog coming out the cells of the perichondriocyte, and these perichondriocytes, they start expressing RUNX2, and under the influence of RUNX2, these cells change into osteoblasts. Osteoblasts are the precursor of the bone forming cell, what we call osteocyte. When they become, there's a lot of wind, we know like there was a lot of wind around, right? When these cells change into osteoblast, they receive wind is in the surrounding. These cells then start expressing ostrix. Ostrix is a transcription factor, and that transcription factor turns these cells into osteocytes. And when they become osteocyte, what they do? They start secreting a matrix, and that matrix is essentially the bone. Okay? So now you have all the bone formation is taking place. So this is the bone. Remember, the bone is hollow. Right? So once these osteocytes are formed, the cells which are right here, the hypertrophic cell, they then hypertrophic cell start undergoing apoptosis. Okay, so these cells, they start undergoing apoptosis, they start dying, right? But then you have to clear those cells so that you can have the hollow thing, right? So then these blood vessels, remember the blood is forming from intermediate mesoderm, we did talk about that. So these blood vessels, okay. So blood vessels bring osteoclast cells. Osteoclast cells they are multi-nucleated cells. They eat up that hypertrophic chondrocytes. So these blood vessels are making osteoclasts, and what they're doing, they're eating up these cells. So what we have this hollow bone is formed. Making sense? So what is going to happen? So ultimately what you will lead to a structure like this. Here will be your osteocytes, and these osteocytes have secreted the bone matrix, and this will be total hollow. And what will you have? You will have these blood vessels there and these blood vessels there. Right? And then this intermediate mesoderm is also going to bring all the stem cells which are going to give you the blood vessels, everything, the hematopoietic cells. But this thing is happening here, but right here, we still have our initial phase. We still have chondrocytes, we still have dividing chondrocyte, we still have perichondrocyte, and we still will form some hypertrophic chondrocyte. Now this plate, this size here, this is called epiphyseal growth plate. Remember that our bones grow till 18, 19 years, right? They grow because these epiphyseal growth plate, they still have the same chondrocytes there, they still have perichondrocytes, and they keep on forming the hypertrophic chondrocyte and keep on growing. Making sense so far? So what happened there? If we talk about the signaling, we are saying the signaling is occurring in two ways. Number one, it's okay to erase it? So let me explain everything so that things are more clear about the signaling. So what we had, in the beginning we had this cell, the mesenchyme cell was there, right? So this mesenchyme cell was there, right? What this mesenchyme cell had, this mesenchyme cell get SOX9 because BMP was there, right? BMP tell it to get the SOX9, and when the SOX9 come, when this cell start expressing SOX9, this cell become chondriocyte, right? This cell become chondriocyte. And then we said, this cell also is getting some Wnt because Wnt is throughout. This cell is also getting some BMP because BMP is also out. Under those influence, this cell become a hypertrophic chondriocyte. Right? This cell become hypertrophic chondriocyte. Now, but this mesenchyme cell, some of these become chondrocyte, the cell which was surrounding it, they become perichondriocyte. This cell become perichondriocyte, this is your perichondriocyte. This perichondriocyte cell was still there, it was still there. What happened? This hypertrophic cell, it secretes Indian hedgehog. Okay? What Indian hedgehog does actually, Indian hedgehog block twist, and actually the twist, it blocks run X2. Now think about it. If the Indian hedgehog is not there, twist will be there. Twist will keeping on block run X2, right? So when Indian hedgehog is secret, it will block twist. So twist cannot block run X2. So these cells, they start express run X2, right? And these cells now become osteoblasts. Making sense? Now, these osteoblasts also receive wind. When they receive wind, they start expressing ostrates. So they start express ostrates, and these become then osteocyte. And then they express bone specific gene. So what we are saying? We said, hey, this endochondral bone formation takes place because first the cartilage is formed, and then cartilage is replaced by the bone. We said it starts with the commitment of the mesenchyme cell. Mesenchyme cells become committed, they become either chondrocyte or perichondrocyte, and they form the compact nodule. That compact nodule is formed under the influence of BMP, that secrete N-cadherin. N-cadherin cause them to come together, and then you have N-cam. N-cam make that compact nodule structure. Then we said next process occurs where the proliferation takes place, the chondrocytes start dividing crazy, and they form a bone module. And then we said during this process, some of the outer cells, they start secreting perichondrium, and then you have the perichondrocytes there. So inner cell become chondrocyte, outer cell become perichondrocyte. Then we said the growth takes place, the inner cell become hypertrophic chondrocyte. Those hypertrophic chondrocytes, they start secreting N-cadherin and collagen 10. That causes calcium phosphate to come there, so the mineralization takes place. Then we said these hypertrophic chondrocytes, they also start secreting in hedgehog. In hedgehog is secreted out on the surrounding perichondrocyte, and what that does, that causes expression of RNX2. And those cells which were perichondrocytes now become osteoblasts. Now osteoblasts can receive the signal, can receive wind, can respond to wind. Wind is already there. When they receive that, they start expressing ostrates, and when they express ostrates, these cells become osteocyte, and they start secreting the bone barriers. While this thing was happening, hypertrophic chondrocytes, they were also expressing VEGF that was attracting blood vessels. So blood vessels started coming in. And when the blood vessels come in, what happened is hypertrophic chondrocytes, they start dying by apoptosis. Then blood vessels bring osteoclasts. Osteoclasts are multinuclear cells which are produced in the lateral plate mesoderm. And what they do, they start eating up those hypertrophic chondrocytes, and you have the hollow formation take place. And then later the hematopoietic stem cell will come from lateral plate, and they are going to be called the blood forming cell. Meanwhile, the center is becoming hollow. The tip are called epiphyseal growth plate. They still have all the prechondrocyte, chondrocyte, everything is there. They aid in the growth under the influence of growth hormone actually. So then they will keep on growing till 18, till all the stem cells. What were the stem cells here? The mesenchyme stem cells, right? They all are gone. Then no more growth of the bone. So epiphyseal plate will have all the chondrocyte, perichondrocyte, and hypertrophic chondrocytes of growth and continue. So this is what we talk about. Any doubt? We should talk to each other for a minute so that things are clear. Not we'll go and we'll end this here.
We just said that it causes expression.
Wait, one more time?
But it
One particular growth plate has perichondrocyte
Everything is clear? Okay, just, if you find me anywhere, anything is not clear, just come and talk to me. Don't worry too much about the exam. Make sure you understand the things, right? And I know this is complex, but this is what it is. But it has direct application to us, right? Okay, so we'll meet tomorrow in the lab and go over the data and decide what to do. Okay? Exam on Wednesday? Okay, good.